some complex analyses such as protein impurity identification and characterization aren't currently feasible in real time. So I want to talk about that real time aspect a little bit. They have to be collected, taken offline or offsite for analysis. If this weren't the case, if these assays could be conducted online and closer to real time, what sort of efficiency gains can you imagine we'd be looking at, Barrett? I'm going to start that question with you.
Sure. So coming from into the biosimilar industry, this was just the most important thing, that ability to demonstrate biosimilarity and interchangeability. So at least for us, at the time, if we were able to measure, let's say, sugar residues, glycan profiles in real time, and to be able to control and pull those levers to modulate and adjust those things, in steady state, you're going to get a higher quality product, you're going to reduce variation, and you're going to know what's coming out without having to wait. So I think that's why I think this, especially for that niche industry, the benefits are huge, being able to monitor those CQAs, because they directly impact how your molecule is going to perform when you inject into a patient. So.
Yeah. We got some more good questions coming in from the audience here. I want to get on to Barrett. Are you comfortable with that auto allo processes question that just came in from Peter?
Oh, are you talking about cell and gene therapy? I'm trying to read it.
What are your thoughts on the PAT development focus in auto versus allo processes? Is one domain more attractive over the other in a business sense from your perspective? So I wasn't sure if that was within kind of your area of expertise or not.
So I do have a few cell and gene therapy clients, and can't mention their names, but they are looking, and they're all allogeneic. So I would just start with that up front, and they are looking to automate a lot of these things. So off the top of my head and based on my current experience, I think allo is kind of more of an opportunity for these PAT technologies, at least at first. It might also make more sense in that, auto is already wildly expensive to begin with, because it is truly personalized. And so if you're looking to add in that ability, that potentially initially ratchets up the cost even more. So allogeneic, I think, is more of an opportunity to implement PAT for continuous, at least initially. That's just kind of my take based on what I'm doing now with folks. So, that's just my opinion.
Paul, let me broaden that question up for your experience at KKNA real quick. Even if it's just within specific to antibodies, is there, I guess, what goes into the business discretion around PAT investment on a specific line? Perhaps a specific molecule, a specific antibody that you're working on. Just give us some color, I guess, on what goes into that decision. Is it where we are on the development continuum? Is it market size? Is it the results of a developability assessment? What goes into those determinations?
Probably yes, and yes. It's all of the above, and I think that's part of the problem, right? You need to make these decisions so early on in the process in order to be able to fully understand the characterization of the product, the CQA elements, and how you can attack those elements. Again, given almost to some degree, given where the science is going to be and what you will be able to measure and control, 5 years from now, right, from a processing perspective. So for me, it's about trying to understand maybe not so much the market size, because the pricing of the product might help to overcome smaller markets. But in cases where consistency of product, in cases where rapid response relative to process parameters are important, there are multiple cases that I'm sure we could talk about where the product needs better processing, more exact processing. So therefore, those process parameter controls and that continuous flow and the benefits that we've talked about, would really drive the success of the product. So I think, from my perspective, it has to do with that speed to market, the regulatory filing, the quality and consistency of the product. Those things are really the ones that are driving. Now, unfortunately, in our industry, what is it, 10, 12% of products that enter phase 2 actually get to live long enough to be commercialized. So unfortunately, there is a good amount of wagering that's going to happen on the upfront process. So that's why, for me, over the longer haul, strategically, this database of capability needs to continue to be developed such that we can continue to develop the process for that 1 out of 10 products that actually are successful.
How much of that, in the event that, in the frequent event that the product is unsuccessful, how much of that effort and investment in product-specific PAT can be recycled, reused? Maybe not reduced, doesn't play, that R doesn't play in here, but recycled, reused, co-opted, leveraged on a following candidate.
Well, I mean, for me, it all of it can, but it's more in the institutional awareness, not unlike research in general, right? I mean, if most research doesn't actually do anything, technically doesn't actually do anything specific, well, then at the very least, we learn some things that then teach us some other things that then allow for, that 5th degree of separation to turn into a success at some point, or at least that would be the hope. So for me, more shots on goal from a product perspective will result in higher probability of success from a product success perspective. It's the same sort of thing from a technology perspective, is the way I like to think about it.